by proper collision with the substrate. When they do collide in the right way, they form base pairs, and then this paired region docks into the active site cleft between the two domains. At that point, if guanosine is present, it can attack the substrate strand, cleaving off, becoming attached to it by transesterification. One of the products leaves very rapidly, and then the other cleavage product, which is held on by base pairing and by interactions between the backbone of the helix and the catalyst, comes off much more slowly. And in fact, in the multiple turnover reaction, which means when the catalyst recycles and each catalyst cleaves multiple substrates, the slow step in this entire cycle, if you have, assuming that you have enough of the RNA substrate to always fill the active site, is this product release step. So it zips through all of these steps, and then there's sort of a slow release of one of the cleavage fragments before you get back to the point where the free catalyst can encounter and interact with a new substrate molecule. Now, the next two slides are a little interlude at a little bit of a more advanced level, because students often ask me, well, what kind of experiments can you possibly do to address these questions? And I find that it's very easy for a scientist to talk to a group of high school students about the results of experiments and what it is that we think we've learned. But it takes a lot more training for students to be able to understand the process by which the experiments are actually done and interpreted. So I'm going to give you a little glimpse into that process. If you don't understand the next two slides, don't be concerned, because I'll return to more descriptive material in a minute. So this is actual experimental data, the sort of which that is generated many, many times per day in the laboratory. That we start out with a radioactively labeled substrate RNA that is uncleaved. The nucleotide sequence of that substrate is shown on the top, and it's radioactively labeled using an isotope of phosphorus called phosphorus 32, which emits beta particles, and only the left end of the molecule is labeled. So that when this substrate is cleaved into products, even though there are two products, you only see one of them, the one that is at the left end, because that's the only one that's radioactively labeled. This radioactive labeling is done because we're working with extremely small amounts of material, and it would be difficult to detect the material by a standard chemical analysis. The phosphorus 32 provides a very sensitive technique, and only very small amounts of the radioactivity are needed because we have very sensitive ways of measuring where the radioactivity is, the simplest of which is to just take a piece of X-ray film like you would find in a hospital, and in the dark room just place the X-ray film over the experiment, come back the next day, remove it and develop it just like you would any piece of film, and then the exposed silver grains show you the location of the phosphorus 32. One thing remains to be described, and that is how do we separate the uncleaved substrate from the cleaved product. And this is using gel electrophoresis, which I described briefly yesterday, that the molecules in the influence of an electric field electrophorese from top to bottom. The smaller ones go faster, and so this provides then the separation between substrate and product. And you can see that as time goes on, in the presence of a ribozyme, this substrate RNA is cleaved to give this product, and that in this particular experiment, it depends on the concentrations of the various ingredients, how fast this would actually take place. But in this particular experiment, you can see that by 10 minutes, there's been essentially complete conversion of the starting RNA to the cleaved products. We then quantitate the amount of substrate and product at each time point and make a graph that shows the fraction of the substrate remaining uncleaved, so that starts out at 1 because it's all intact. We plot the amount of substrate remaining as a function of time, and you can see that depending on the amount of guanosine, remember guanosine's the little scissors that are cleaving the RNA, depending on the amount of guanosine present, we either get this straight line, or at a higher amount of guanosine we get this line, or at an even higher 3 millimolar amount of guanosine we get a faster rate. And then from the slope of each of these lines, and many more lines that I haven't shown you, we can make a plot then of the rate of the reaction as a function of the guanosine concentration. And this shows a behavior which is very similar to what a protein enzyme would show. Namely, at low concentrations, the rate increases proportionally to the guanosine concentration. But then you come to a point where the rate is constant. You get to a maximum velocity, in this case, about 8 times 10 to the minus 2 per minute, which no matter how much guanosine you put in, you don't exceed that limiting or maximum velocity. And that's because at that point, the guanosine binding site is always full. And so adding more guanosine doesn't help the reaction because there's enough guanosine in solution to keep that guanosine binding site occupied. So from this sort of an experiment, and others not shown, we can get an estimate of the affinity of the ribozyme for guanosine, and we can see that it's binding guanosine, it's half occupied at about 0.5 millimolar guanosine concentration. So this gives you a bit of a glimpse of the laboratory procedures that one would have to go through in order to evaluate a catalyst, in this case an RNA catalyst, but very similar methods would be used for evaluating a protein catalyst. I'd now like to turn to the question of how is the rate acceleration achieved, and a substitution of a single atom right at the cleavage site. Experiments done by Joe Piccirilli, a former postdoctoral fellow in the laboratory, have been very revealing in this regard. We compared the cleavage, so this is the phosphorus atom at which the guanosine attacks. And so this is the bond that's actually going to be broken in the first step of the self-splicing reaction, or in this RNA enzyme system that's analogous to the self-splicing reaction. We compared the activity or the rate of cleavage with 2 different atoms in position X. Oxygen would be the normal occupant of that particular site if this was regular RNA, or in this case it's actually, for ease of synthesis, we did this as a single-stranded DNA molecule. There's no hydroxyl group in this position. And the ribozymes will cleave single-stranded DNA, although they do so much more slowly than they cleave RNA. We compared then the cleavage of the oxygen-containing substrate with a 3' sulfur substituted substrate. And you know from your studies in chemistry of the periodic table the relationship between oxygen and sulfur, right? How are they related to each other? Somebody remember? They're in the same group, so they're very good. So they're one right on top of the other, which means that they have very similar chemical and bonding properties, but they have differences as well. And in this case, we substituted sulfur because from a chemist's point of view, that should make it much easier to cleave the bond between that atom and the phosphorus because the negative charge that's going to accumulate on this atom when the bond is broken is going to be more easily accommodated by the sulfur than the oxygen. And so this should speed up the reaction a lot. And when we did the experiment, we found that the normal phosphate-containing substrate was cleaved to completion as expected, and completely opposite to expectation, this 3' sulfur-containing molecule was not cleaved at all. And so this was a disappointing result because it